I'm excited to tell you about some of the work that I've been doing recently, as well as into the past. And what I'll talk primarily about today is malaria and malaria parasites and how they evolve. But we're going to use malaria as sort of a nice thread to introduce some ideas about generative models and multi-layer networks as we go. And we'll branch off but eventually come back to the main story. So this is just a very disappointing map. This shows Plasmodium falciparum, the malaria parasite, prevalence among 2 to 10-year-olds, so these are kids. And this is data for the past 5 years. As you can see, despite the fact that we've made a lot of progress in fighting malaria around the globe and reducing prevalence, this is still something that causes hundreds of millions of cases annually and kills around 500,000 people each year, mostly kids. So malaria continues to be a problem, and one of the reasons has to do with the fact that malaria parasites live outside the public health infrastructure. Some of my work was done in Kenya, and the neighborhood just outside of town looks basically like this. There's not a huge road infrastructure, there's not a great public health infrastructure. The hospital that I was working at, this is a photo of the orthopedics department. It's not exactly a strong-looking orthopedics department. And one of the reasons that I'm showing you orthopedics instead of parasitology or infectious disease is the fact that a lot of the folks were waiting outside those other departments, so I didn't want to take a photo of them for privacy reasons. The other reason malaria is hard is that the parasites live inside red blood cells. So these are stained red blood cells, and so the round things here are the cells, and if you see these stained dots in the middle, those are parasites that are replicating. So the parasites live outside the public health infrastructure. They live inside blood cells, which means to study them in the lab, we essentially need to feed them blood, or feed blood to them. They're sort of reverse vampires, right? And as a result, they're very difficult. But as I'll tell you about today, I think networks are giving us a little bit of an advantage in learning how these parasites evolve. And I think that networks will be among the tools that actually help us to crack malaria with help from other more direct interventions like MedMeds. So my work in general focuses on the mathematics and methodologies to understand how malaria's genes evolve, both at the scales of individual parasites and populations. And today, I'll introduce malaria's solution to what we'll call an arms race in humans. Then we'll talk about generative models for networks, in particular community structure and ranking. And then finally, multi-layer networks, and we'll talk about some key concepts, which are interdependence, versatility, and resilience. I'm just noticing now that the slides on the screen are a little bit cut off, but on the monitor here, they're full. So if you see something cut off, check out the middle. To talk about malaria, we have to talk about the parasite life cycle. And it is a cycle, right? But we're going to arbitrarily cut it and say that it just begins when a mosquito that's infected bites you. And this mosquito bite introduces parasites, and these go into the liver and they replicate. They emerge at some point en masse from the liver and enter this red box here, which is the blood stage. So each individual parasite is going to find a home inside a red blood cell. And when it's inside the red blood cell, over the next 48 hours, it's going to multiply from 1 up to between 15 and 20. All of the parasites in synchrony inside their individual red blood cell homes are going to emerge en masse. And then each one of those individual parasites finds its way into a new home, and the cycle continues. So if you have had malaria, or if you know somebody who has had malaria, one of the characteristics is what's called a tertian fever, meaning that every 48 hours you get this fever spike. On the third day, you get the fever spike. And this continues when all the parasites burst out and your immune system goes nuts. And then they find new homes and you sort of recover a little bit, only to have it recur the next time. But eventually, some of these parasites stop just going through this replication. They form sexual stages, they get close to the skin, and then a mosquito bites a human, gets the parasites, and the cycle can continue on the mosquito side. What we'll focus on today is this blood stage. I'm showing you 2 artificially colored on the top and no color on the bottom electron microscopy images that show infected red blood cells. What I want you to notice is that these red cells here that are colored red look like our standard textbook picture of what a red blood cell looks like. It's sort of like an inner tube, right? Nice and round, smooth looking. An infected cell looks like this. It's mottled, it's deformed, and in fact it's very brittle. And one of the reasons is that the parasite is sort of remodeling its own home. What it's doing is it's expressing on the surface a sticky protein. This protein works sort of like Velcro and allows the parasite to sequester all around the body instead of freely circulating. So what you can see in this bottom slide here are infected red blood cells that are stuck to an endothelial cell, and so they're stationary, they're not circulating. We're going to study that Velcro. It's the product of what I'll call an evolutionary arms race. So the spleen mechanically filters your blood. So if you have a very brittle parasite inside the red blood cell, it would get filtered out. So the parasites avoid free circulation by using these sticky surface proteins. This is a process we call sequestration. But the immune system is going to see that Velcro on the outside of these infected red blood cells. And so it targets those red blood cells. So what the parasites do is they just express different camouflage patterns in those surface proteins. We go from Velcro version A to Velcro version B, and this confuses the immune system, which then needs to catch up. We would call this immune evasion through antigenic variation. And just to decode this, an antigen is just something that's recognized by the immune system. So antigenic variation is this process of sequentially shifting the camouflage patterns on cells. So over time, of course, your immune system accumulates knowledge of things that it's seen in the past. So immunity to these various camouflages will grow. And so the parasites shuffle their VAR genes. These are the genes that are responsible for creating this Velcro camouflage in the first place, making new patterns. And this is what we would call diversifying selection through genetic recombination. This is not survival of the fittest, it's survival of the most diverse. The problem is that this very same process that enables the parasite to survive, namely shuffling its genes, also disables standard forms of analysis. So let me explain the mechanism just a little bit, and then we'll get to where networks actually come in. So each parasite has essentially a deck of cards worth of VAR genes, 60 different varieties in this case, and I'm just representing those by different colored lines. But actually each VAR gene is composed of a variable number of domain classes or blocks, and I've colored those different classes accordingly, and we have just a various mixture of different blocks from each of these genes. I'll just point out that the first domain is usually of the same class, and so that's why I've colored all the first ones. Okay, same class in a statistical sense, but still in the genetic frame-by-frame sense, widely different content. So I'm just representing that in the picture by different patterns on the blue blocks. So these genes evolve by recombination. So I think everybody in here probably has a decent understanding of the basics of biology, right? We have mutation, where the base pairs change letters. We have insertion, where we get new base pairs. We have deletion, where we remove base pairs. And this is sort of the basic way that we learn about how evolution works. And over time, you accumulate mutations, and a parasite, or a human, or whatever can adapt. In this case, we have a fundamentally different mechanism. Recombination looks more like we take two existing genes, we cut them in half, and then we end up with something totally new. So this is not small-scale little iterative work, this is large-scale recombination. And not only can we get these sort of simple, let's just cut once things, but we can get what are called multiple crossover events, where I end up with something that appears to sort of randomly alternate between the two progenitor genes. Not only that, we can also make cuts in the different domain blocks along the genes. So here, if I cut in the blue, and I also cut in the gray, and I make a new one, notice that this new one has a different combination of blocks than either one of the parental genes did. So this fundamentally different mechanism introduces problems. The problem is that trees, which are really the workhorse of phylogenetics, assume evolution by mutation, insertion, and deletion, which are fundamentally bifurcating processes. So when I say they're bifurcating processes, what I mean is that, let's say that we have a pool of genes, and they're all the same, right? And then one of them develops a mutation. So we can now split the population with the mutation and without the mutation. Now let's say that in those genes that have built up, that have the mutation, we have a new mutation that comes in. So now we have a new mutation as well, and then just the old mutation. So this is a splitting process, right? So if we have genes and a pool of genes that are all evolving by mutation, insertion, and deletion, and we fit to a tree, then fundamentally, the tree is the right model for the data. However, if we have genes that are recombining, shuffling, then trees no longer actually fit the data. So if you try and fit these VAR genes using trees, you get trees that look like this. Everything is connected at the root. You have no understanding of what the population structure is here. So essentially, applying tree methods to VAR genes gives us garbage. in the way that we expect when the model and the data are just fundamentally mismatched.
Yeah, is this unique to malaria, or do you see this in lots of other?
So there are a lot of organisms in which we see recombination. We see things like horizontal gene transfer. We see things like plasmids and other cool things that are sort of not mutation, insertion, and deletion. Malaria just happens to do it much more rapidly. And for these genes, this is not just sort of like an additional mechanism that sometimes occurs in addition to like mutation, insertion, deletion. In this case, the recombination is the fundamental process that the parasite is using to evolve and evade the immune system. Yeah. Okay, so if we want to understand these genes, we need some kind of analytical framework that's actually appropriate for the mixing and shuffling. So what we'll do is, surprise, develop a network-based method. We're going to focus on the DBL alpha domain, which is present in almost all of our genes. That's this blue domain here that I was talking about. We like the DBL alpha domain because DBL also happens to be my initials, so I feel like a certain closeness to the problem. So this protein, this Velcro, it needs to evade the immune system, but that's only its secondary characteristic, right? The primary thing it needs to do is to be able to stick to the endothelial cells around the body. So for that reason, some of the backbone structure needs to be preserved across variants. So what we see is if we take hundreds or thousands of these genes and we do an alignment and we line them up, we see this alternating variable, conserved pattern. In other words, they're highly variable regions shaded in gray, and spacing them are these conserved sort of backbone elements of the protein. So if we're trying to understand how the parasite is bobbing and weaving and evading the immune system, we don't even care about these conserved parts. These are just for binding. What we want to look at are the highly diverse parts that the parasite is using to sort of throw off the immune system. So what we're going to do is map sequences to complex networks with that variable region idea in mind. So here I'm just showing you some cartoon version here, a bunch of sequences from one parasite, 4 sequences from another, 3 from a third, or some number from a third, some number from a fourth. And what we're going to do is essentially line up, take out those conserved regions to find the highly variable regions, or HVRs, in between. And at each one, we'll build a network. I'll talk a little bit about data for this study, but here we're using 7 parasites, genomes from around the world. In total, we're going to have 307 sequences, so 307 bars here. And we found 9 highly variable regions. In the cartoon I'm just showing you before. So in each highly variable region, we're going to map sequences to an all-to-all connected and weighted network. And so what we do is, each node in the network will correspond to an amino acid sequence. I'm going to connect 2 amino acid sequences with an edge whose weight corresponds to the longest shared substring. So if you look at the bottom, you look at the top, you can see there's a substring here of length 16 that's shared with the top. And so those 2 nodes that correspond to those genes are going to be connected with an edge weight of 16. So I do this for all of the possible combinations, and I end up with this all-to-all connected weighted network. The idea here is that a long shared substring implies a recent recombination or some hereditary relationship between 2 of the genes. So essentially, I'm connecting genes that are similar, and the more similar they are, the stronger the weight is. We're then going to threshold the network, which in general is not a great idea. In other words, we're going to remove all the short substring edges and then discard the weights. So I'm going to take something that's very rich. It's got all of these edge weights in it, and it tells me how related 2 sequences are. And I'm going to say that if you're above this level, then I'm going to put a 1. If you're below the level, I'm going to replace it with a 0. And in general, this is not a good idea, right? We're taking a very rich data set, and then we're like hitting it with a very stupid hammer and turning it into a bunch of 1s and 0s. In this case, it's sort of an active decision. One of the reasons is that the biological difference between a length 8 substring and a length 16 substring is entirely unknown. Is a shared length of 16 somehow like twice as important as a shared length of 8? Should we scale things logarithmically? We don't have any sense of that. So instead, what we're going to settle for essentially is these are related, and then if it's too short, then they're not related. Our goal then is going to be to find community structure within these networks. So now that we've taken all of the genes and we've connected them, We're going to choose a method for community structure that's congruous with our prior beliefs about the system. So namely that our goal is to find population structure. So our hope is that genes can be divided into latent groups that somehow reflect restrictions on recombination. So let's say that I have one big pool of genes, and now I say that there's a restriction and that I'm not going to get recombination across this barrier. What I would expect then is more links within these communities and fewer links in between. And that's sort of the premise here, except we don't know what those communities are a priori, and so our goal is to discover them from the data. But we also want a method that's going to be okay when we know that we're only observing a subset of the vertices. I only have 307 sequences here, but in the wild there are 10,000, 20,000, who knows how many. We also know that edges may be missing or spurious. This could be due to sequencing errors. This could be due to the fact that we're imposing a threshold. So what we're going to use for community detection is something called the stochastic block model. The stochastic block model, or SBM, just models the probability that gene I and gene J share a substrate. It says that probability is only determined by the groups to which they belong. So if I and J are in the same group, we're going to have some probability they connect. If they're in different groups, we're going to have a different probability they connect. But I want to dig into this because the stochastic block model is what we call a generative model for networks. And fundamentally, it's a different way of thinking about community structure networks than some of the other methods that are circulating. So a generative model in general is just a recipe. We choose some parameters, and then we make a network. So here I'm showing you a ring network, and you can imagine the recipe that we might write this. Let's say I tell you the number of nodes, and I tell you how many links each one is going to have, and then you just arrange them in a circle, you connect the adjacent ones, and maybe the bi-adjacent ones, and so on. Choose the parameters, follow the recipe, get the network. A stochastic generative model is also just a recipe. It's just that now there's some randomness involved. So we choose parameters, and then we draw a network, not necessarily the network. So if I draw, again, from the model, I might get a slightly different network, but presumably, based on the parameters, I'm going to end up with something from the same sort of class or family of networks. So since a single stochastic generative model can generate many networks, the model itself corresponds to a set of networks. Right? When Ole was talking, he was mentioning that in this coin flipping game, you might get a good trajectory that you like, you might get a bad trajectory. The model itself corresponds to the totality of all those trajectories. In the same way, a generative model for networks corresponds to the whole set of networks that it can produce. But since this generative model is a combination or a composition of random variables, a random graph model, a random generative model, is a set of possible networks, and each one has some probability associated with it. There may be some networks that we create with high probability, and other networks that we create with low probability. So to give you an example, if I talk about a random network where I take n nodes and I throw down m edges, it's very unlikely that I end up with a perfect ring, right? It's also very unlikely that I end up with a clique where I have all of the edges connecting only very few nodes. On the other hand, there may be some networks that are created randomly that have a higher probability than others. So our goal with real data is essentially, if you'll pardon me using this metaphor, we're going to find a balance of ingredients in the recipe that's most likely to have generated the network that we actually have. So in math, here's what it looks like. We're going to define a parametric probability distribution over networks. So the probability that we get a graph G conditioned on the parameters in theta. Generative models are cool because we can generate data. So if you give me theta, I can draw lots of graphs by just flipping the coins or however I'm generating randomness. And I can draw from this distribution P of G conditioned on theta. So that's taking the model and then generating a graph or some set of vertices and edges. Then we can also go the other way, and this is statistical inference. So given G, an actual network, choose the theta that makes G more likely. So this is going from data back to the model. And in the process, when we do this inference, we learn these parameters theta, and therefore we learn a model-based description of the network. To put a little bit differently, if I have something like the Zachary karate club, I have some network that I think has community structure in it. I can write down over all the possible divisions of those nodes into different groups, what's the likelihood or what's the probability of having generated that network given that division into groups. And so if I have a crummy division into groups, I have a low probability. If I have a great division into groups, I have a nice peaked probability. And so the goal in fitting a generative model in the inference process is essentially to find your way through this space until you find the correct parameters, or the most likely parameters to have generated your data. The general form of one of these is that the probability of G given theta is some product over all possible edges, I and j, of just the probability of edge aij given theta. And the only reason I'm writing this down is to say that we're going to draw each of the edges independently. That is, if I'm trying to write down a network for the people in this room, I'm going to consider every single pair of us independently and flip coins or do whatever other process to generate those edges. This conditional independence is really important. And all the assumptions about structure itself go into this probability. So this is really the edge generation function, or the recipe. One of the keys of this is consistency, meaning we want to close this loop between generation and inference. Namely that if I generate an infinite amount of data from the model, I'd better be able to exactly correctly recover the parameters, right? Like, if I take an infinite number of samples from here and get lots of data, I'd better be able to always recover exactly the parameters that I planted. If I go from the left to the right and then right back to the left, and I don't end up where I started, then it's really not a good model. And as it turns out, this conditional independence, this general form on the top, is the only way that we have some hope of getting there. So there are two main categories that I'll tell you about. One are stochastic block models that we spoke just a little bit about, and the other are latent space models. So let's dig into these. Stochastic block models, we basically just divide the vertices into groups. So let's say I have k different groups. In this picture on the right, k equals 3. The probability of an edge between I and j is going to be dependent on, now I'm introducing the parameters here, m is going to be our matrix that tells us how the different groups relate to each other. So if there are more connections within a group, I want the diagonal entries of m to be high. And if there are fewer connections between different groups, then I want the off-diagonal elements to be very low. This says there's a high probability of connections within groups, a low probability of connections between groups. Doing so gives me something like the network here, where the blues tend to connect to the blues, the greens to the greens, and the reds to the reds. The point here is that the probability that two nodes connect depends only on their group types, not on other factors, not on hair color, or whether or not they have glasses, or whether or not the nodes are widely parasites or stupid parasites, right? Okay, so this was originally invented by sociologists, and there are lots and lots and lots of different flavors here. If you look at the slides online, I've got links to all of these. So where does the name stochastic block model come from? It comes from the fact that if I take a network and I fit it to data and I divide it into these groups, and then I look at the sorted adjacency matrix between these, what I see is that there's a high probability of connection within these groups, low between the groups, and you can see that it sort of naturally sorts itself into blocks. Essentially what I'm doing is I'm creating random matrices in each one of these blocks. It's just that the parameter of that random matrix happens to be different depending on whether or not you're within the block or whether or not you're in a different block that corresponds to different groups. So this is kind of cool because it means that we get a lot of diversity of the kinds of structures. So we can have assortative communities. So these are lots of edges within groups. You can see that we have a dominance on the diagonal here. And I don't know if you guys can see, but we have the green connects to the green, the pink to the pink, and so on. But very few connections in between. But we can also have disassortative communities. So now I'm saying you don't get to connect to people within your group, but you do want to connect to people in other groups, and we can get different structures. Of course, visually, this looks like garbage down here. We can't see the structure. But when we actually look at the blocks themselves, the structure just pops out. We can also have ordered group structure, where you tend to connect to people in your group and in adjacent groups, and then you get sort of a linear group hierarchy, or core periphery structure, where the core connects to itself, the periphery can connect to the core, but the periphery never connects to the periphery. So you get this diversity of structures just depending on the parameters that we plant, which means that if you give me a network that happens to be generated by any of these, we can extract that structure through statistical inference. Another kind of generative model is called the latent space model. The idea here is that nodes live in some latent space. So we're going to embed them in some kind of imagined line, or imagined Cartesian plane, or imagined hyperbolic space. But in this case, the probability that I connects to j is some function of the position of I and the position of j in that latent space. So this is something that's been super useful in not finding community structure, but instead finding hierarchy. So if I give you, for example, all the hiring patterns between universities and which universities hire which other universities' PhD students, you can apply these latent space models and essentially find an ordering of prestige hierarchy. If I give you a food web, you can do the same kind of thing and find which are the apex predators and which are, the bottom feeders, essentially. You can also use this with sports teams. You can use this with online dating. And in fact, I don't mean like as a strategy for online dating. I mean for if you're developing an algorithm in your online dating startup that's trying to present one person with some good matches, you might say, well, if somebody is right in the middle of desirability space, I should either present to that person people who are very far away on the low side or very far away on the high side. I should present this person with somebody who's of around the same desirability. And in fact, Tinder uses something like this, I'm told, in how they present people, who they're going to swipe through. Fine. So there's some opportunities here, there's some challenges. I think I'll mostly skip this, and if you're interested, you can ask me about these opportunities and challenges. You can also see these on the slides that will be posted online. But in general, generative models are a very powerful tool because they're rooted in statistically principled techniques. We don't accept generative models that can't close the loop, that can't infer the parameters from the data that they generate. One thing that I want to emphasize here before moving on, though, is that we can learn from partial or noisy data. And this is key for a reason for choosing it to analyze malaria parasites. We can extrapolate from data, we can interpolate, we can infer missing data, we can infer hidden data. We can also do all these things in the presence of noise. And those statistical guarantees that come up in generative models are super, super attractive to this. Okay, also there are problems, but ask me about this if you'd like. So back to the problem at hand, I was arguing to you that we should use the stochastic block model because its assumptions are really, really nice given the kinds of questions we want to ask about this biological system. So, a problem. We visualize one of these networks, which I think is always a nice sort of entry point into a problem. You can see a problem right away, which is there are these all-to-all clusters, or cliques, embedded within the network. Here's one, I've kind of circled them around here. So these are kind of blobs within one of these networks. And the key issue is, again, model data mismatch. Right? I was complaining about trees and saying trees are not good for these genes because the data are fundamentally different. Cliques are very low probability under most community detection models. For example, modularity struggles with cliques of different sizes and hits what's called the resolution limit, particularly when cliques are embedded within a larger network, which is exactly what we have here. Stochastic block models also bad performance. They create cliques in the generative process with probability approaching 0 as you make the clique bigger. And the reason for that is simple. In a random network, the probability of creating a clique drops as the size of that clique increases. So one question that we have to ask here when we're confronted with model data mismatch is, why are there cliques in the first place? And of course, we're probably all on the same page here. The answer is Kevin Bacon. So you've probably heard of this game, 6 Degrees of Kevin Bacon. I'll name an actor, and you have to find some path through movies that connects to Kevin Bacon, and you have only 6 chances or 6 steps to get there. So for example, if we're talking about Bono and Kevin Bacon and Charlene Woodward were in He Said, She Said. Bono and Charlene Woodward were in the $1 Million Hotel, and so from Kevin Bacon, in 1-2 steps, we can get to Bono, okay, if the dream come true. So what this is saying is that we can have this nice path, these hops, in 2 steps. But I want to point out something that really, we're doing more than 2 hops. We're going from Kevin Bacon to He Said, She Said, to Charlene, to the $1 Million Hotel, to Bono. We're hopping back and forth between actors and movies. So really, this is what we call a bipartite network. When we look at 6 Degrees of Kevin Bacon, we're hopping from actor to actor through the movies, but we're not acknowledging the movies. There were other people in the $1 Million Hotel, right? So if you imagine taking this bipartite network and then going back into the unipartite space where I'm just connecting actors, what I would do here is I would connect Bono to Mia Jovovich to Mel Gibson to Bono. In other words, I would create a triangle. But there are a lot of people in the $1 Million Hotel. So if I take this bipartite network and then I go back into the actor-actor network, what I do when I do this is I create a big clique. Bono connects to Mia, Mel, Jimmy, Jeremy, and so on. They all connect to each other. So Vargine networks, as it turns out, are one-mode projections. Right? We have genes, that's what I started with, and we had substrings, these are the boxes here. And when I'm taking this bipartite network that connects genes and substrings, and I'm just writing it as a gene-gene network, I'm implicitly creating a projection of this bipartite network. When I write this down, it's taking this whole network and just projecting it as a gene-to-gene network. We have lots of examples of these, right? Plants and pollinators, bipartite networks. Papers and authors, bipartite networks. Documents and words, actors and movies, social network users and their mobile access locations. The fundamental thing about a bipartite network is that you have 2 different types of nodes and you only have connections between the types, but never connections within the types. So all co-authorship, co-starring, or co-whatever kinds of networks that you may have thought about are fundamentally one-mode projections. So in this case, we have avoided model data mismatch by going from trees to stochastic block models, but now we have to solve a new problem, which is we have a bipartite network. So we want to solve this bipartite community detection problem directly, rather than projecting and solving the community detection problem in the projection. And at the time, there was a gap in the literature. We didn't have a generative model for bipartite networks. So we'll write down a bipartite stochastic block model. And again, back to the recipe metaphor. What we need to do is write down a recipe that creates bipartite networks. And if we can do that, then we can infer community structure in bipartite networks. And the key thing is that we always know in practice whether or not you're a plant or a pollinator. Right? There's never a case where we have a bipartite network, but we don't really know that information. And if it's a bipartite network, it's immediately recoverable just by trying to color the node, which is on which side. So let's make use of that data when we actually solve this problem. Yeah?
Is it your choice which one's subject, which one's object? You know, authors are papers, papers are authors.
Yeah. So you could
question you want to ask. Or
yeah. So you could take an actor movie network, and you could have these co-starring networks if you project one side. But you could also have these sort of like cast sharing networks if you project on the movie side. Okay, blah, 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 we can write down this probability. But what I want to point out here is just one key thing, which is that when we solve this problem, we embed in the generative model exactly what we want, which is something that can create bipartite networks, and that's in this parameter t, which comes up in the product. I don't want to exactly translate this, but what I really want to say is that when you write down a generative model for a particular kind of data, you can perform inference on that kind of data better. But if you write down a generative model that creates data type X, and then you try and use it on type Y, you're not going to do very well. And so this is the fundamental strength-weakness of generative models. If you've got the generative model right, you're going to be hitting home runs, and if you get the generative model wrong, then your inference isn't going to be necessarily optimal. Okay. So this is kind of fun because we have all kinds of different networks, so we could test this bipartite community detection on exactly the kind of network that I brought up, one of these actor movie networks. So I downloaded IMDb for 6 years. I mean, I got 6 years of data, but the download was fast. I chucked out all one-movie actors and one-actor movies. Sorry. You end up with about 100,000 nodes that's not quite evenly split between actors and movies. And then you can choose the number of groups that you want. In this case, I asked for 6 on each side. And you can find community structure very easily within this actor-movie network. And so here I'm showing you the sorted adjacency matrix, and you can clearly see the block structure here. I've added some labels by additional metadata that I could download from IMDb. And if like 90% of all the check films were in one box, I labeled it check. So you can see some of these labels here. And they tend to be partially explained by these movie metadata, language and genre. And what we're really seeing here are the choices that the consumers push onto casting directors, where we're seeing like which actors tend to be in which kinds of movies. One interesting thing that I'll point out is that adult movies cluster by themselves. And the aspect ratio is totally different here. Specifically that one actor will be in far more movies in the adult genre than in other genres. So the mechanisms that are creating these different blocks depend on the metadata. They depend on the kind of movie that you're looking at. Okay, but back to malaria. So we wanted to use this network methodology to probe VAR gene evolution. So again, we have data from 7 parasites, 307 sequences. We found 9 highly variable regions. We built 9 different networks. The reason we build different networks is that different regions on the gene correspond to different parts of the protein. And different parts of the protein are going to be pushed on and influenced and culled by the immune system in different ways. So we want to allow for the fact that these could be evolving independently. We identified communities separately for each highly variable region using the bipartite stochastic block model. An example of this is shown here. The genes are the big circles, the small circles are the substrings, and you can see we divided here into 3 groups. 6 of the 9 highly variable regions exhibited highly non-random recombination patterns. In other words, they actually had some community structure there. 3 of the 9 had no discernible community structure. So we can ask a fun question. We've looked at 9 different locations on the gene. 6 of them seem to be meaningful. We can ask, is the structure consistent across these highly variable regions? This tells us something about evolutionary constraints, because if the constraints are the same at 2 locations, then we expect that when one location evolves, the other location has to evolve with it. So we'd expect that the structures that we're creating in those are moving not necessarily in lockstep, but let's say noisily together. We would therefore expect that the structures that we get in terms of communities in one location and in the other location should be similar. If the constraints are different at 2 highly variable regions, we expect different large-scale structure. So they're able to move independently, and therefore they're exploring sort of different antigenic spaces. If evolution is unconstrained, in other words, it's totally random, then we expect weak or no structure. So what we can do is use something called variation of information that's sort of like asking how far apart are 2 different divisions into communities. And if I show you the distance here versus the highly variable regions, I'm only showing 6 here that were meaningful, you can see that in general, they tended to be far apart, except for 1 is close to 6, and of course, reciprocally, 6 is close to 1. So what we're arguing here is that 4 of these regions exhibited different but non-random evolutionary constraints, whereas 1 and 6 are coupled in some way. They have similar constraints, and they need to move together. So this gives us a hypothesis for how the parasite is actually evolving. So these highly variable regions enable parallel and partially independent VAR evolution. So most of the HVRs are structured. In other words, they're non-random, but they're moving independently and they're evolving under independent constraints. And the one reason that you might want independent function is if you just want diversity of what you're presenting to the immune system. That's what you're optimizing. It's going to be good for immune evasion. But other regions, like 1 and 6, are moving together and they're coupled to each other. This means that there are similar or coordinated constraints that are required of these different regions. And so some reasons that you might want that in your protein if you're the parasite are endothelial adhesion, you want to be able to actually stick. Or you want to create some particular protein structure. And if you move one part without moving the other part, then suddenly the protein falls apart. It's useless to the parasite, and so on. So we've applied this in a few different cases, other than just sort of some theoretical studies of genes. Humans aren't the only ones to get malaria. In fact, primates of all different kinds, monkeys and great apes get it, rodents get it, birds and reptiles get it. And in this case, we sequenced blood and fecal samples from free-living gorillas and chimps, using techniques that target VAR genes in their blood and in their feces. And the question was, what is the evolutionary origin of VAR genes? Like I've told you this story about how they're bad for humans, and it's part of the survival mechanism in humans, but as it turns out, we were able to find through the network analysis that the genes originated back here at this branch of this subgenus called Larremore, which infects chimpanzees, gorillas, and humans. Which is to say that this mechanism of evading the immune system did not evolve to make us sick. It evolved to survive in our cousins, basically, before humans even speciated from chimpanzees. So it's kind of a cool story here. In fact, it may be the case that Plasmodium falciparum causing such terrible illness in humans is maladaptive, and it's only because of the rampant success of humans in colonizing that it's been able to sort of survive. Killing your host is usually maladaptive for parasites. Working with colleagues in Dakar, Senegal, we're trying to understand more about malaria elimination, and to do so, again, we're using these network approaches. In this case, we've been sequencing parasites and collecting immune cells from people who have been infected over 9 years across this area called Chess, Senegal, which is just inland from Dakar. We're trying to learn how the parasites are adapting as a population to a changing immune landscape. One of the weirdest things about malaria is that if you get infected and you develop some amount of immunity, that immunity wanes over time. So if you're then reinfected 3 years later, it's almost as if you've never been infected before. So what that means is that as the parasite is evolving to evade the immune systems of everybody in the population, the immune systems of everybody in the population are both evolving to respond to parasites and also forgetting the lessons that they've learned. So if you're thinking about evolution on some kind of adaptive landscape, you have evolution, but the landscape is also changing. It's a bit of a mess. Okay. So in the last section here, I want to point out the fact that we looked at multiple networks in this case, right? We took this gene and we found 9 locations on it, and we built a network for each one. And analysis of these malaria parasite genes suggests this layer interdependence, right? HBRs 1 and 6 were somehow coupled to each other. So we can ask whether or not we have multiple networks, or whether or not we should think of this as one multi-layer network, right? We have the same genes, we have the same sequences in all the layers. We're just looking at different locations of those genes. So in what sort of circumstance would it be beneficial for us to think about this as 9 different objects versus one object in which we just happen to share the nodes? So now I want to tell you about multi-layer networks. In single-layer networks, we know that we have nodes and edges, or vertices and links, or whichever vocabulary you like. In multi-layer networks, we have nodes, edges, and layers. And the rule is just that now the edges are going to capture different types of relationships. We can express this as just different colors of edges. Each layer contains all edges of one type, and nodes are the same nodes in each layer. So for example, think of the social networks that you belong to. Maybe we follow each other on Twitter, but we're not friends on Facebook. Maybe we're friends on Facebook, but we never message on Instagram. Right? Okay, those are super cheesy examples. But some more reasonable ones are things like air travel. So this was a study where they looked at the air travel network in Europe, and you can see like what Ryanair's network looks like. Lufthansa, obviously very focused in Germany, Vueling, British Airways, and then the aggregate network. And there are a lot of cases where we're really only studying the aggregate network. But in fact, we could break a network apart into many, many layers. Or where we're studying one layer at a time, when in fact we should be looking at them all together or in aggregate. And I want to point out something, which is that when we were traditionally booking with airlines up until the internet, we were essentially just like going one at a time through these layers. And the sort of disruption that groups like Kayak, or maybe in the past travel agents brought, was really looking at this aggregate network where you're allowed to take one hop on British Airways and then another hop on Lufthansa. I mean, this is also the purpose behind the various airline alliances, which is that they want you to be able to jump from one layer to another, but only if you're part of the same club. There's a really nice study that brings up a different aspect of these, which is that it's not always the case that you can get from a node in one layer to the corresponding node in another layer. So in this case, some researchers got 5% of the samples of Oyster cards in London's transit. People can take the tube, they can take the DLR, which is like the
Docklands Light Railway.
Docklands Light Railway? Okay, yes. Another kind of train. Or the overground. So they have different numbers of nodes and edges in each one of these, and you could look at the aggregate network. You could also ask something about navigability of this network. So because you see the Oyster cards, where people go in and where they come out, you have weighted edges. So you know how many people have gone through each station. So navigability is determined in part by the interlayer links. Where can you actually make the transfers? For example, King's Cross, you can get on the tube, but you can also get on the overground. So this brings up a key concept that I want to point out, which is called versatility. So you've heard about centrality, when I can ask which nodes are central or important, which are the hubs to which information passes. Versatility is a similar concept, but instead of thinking about which are the nodes that are important for moving around in a single layer, I'm going to ask which nodes are important, particularly as gatekeepers or bridges between the layers. Robustness also comes up in these systems. So maintaining navigability in spite of link or node failure is robustness. And we can think about topological robustness or dynamic robustness. So let me explain this too. So topological just means structural. So if I start like punching out links, or if I start punching out nodes, how well can I still navigate the network? So let's say I take out King's Cross. I say like, it's maintenance, right? This is like the story of New York or Boston subways, right? We're gonna repair this line for a while, or like, this station is closed for the next 3 years. Can you still navigate the network? I mean, this is a fundamental question. Can you go in, can you go in at Boston Logan and still make it to Cambridge? Maybe. But as you remove more and more of the nodes, the network falls apart. Dynamic is the same kind of question, except now, instead of just asking whether or not the network has fragmented into multiple components, we're instead going to ask whether or not if I put a random walker on one node, whether or not that random walker can fully explore the network, and how long it takes the walker to fully explore the network. So again, as we remove edges or we remove nodes, the networks can fall apart in different ways. And if you are an engineer and your goal is to maintain a nice system, maintain a system that is robust to certain kinds of failures, then thinking about these issues, particular versatility and robustness, are going to be very important. What was nice about this study was that they didn't do, as Peter called it, the I'm a physicist and I'm here to help thing. They looked at real kind of situations. So in reality, we don't lose like one segment on the MTA. We lose an entire line for an afternoon. So partial line failures rather than just single stations or single stretches. So we talked a little bit about community structure. And we can also think about community structure in multi-layer networks. There are 3 key approaches that I want to highlight. So one is non-generative. You've heard about modularity maximization, discovered by Michelle. But in this case, we can take modularity maximization. We can take this idea of clustering together nodes that are in the same group. And what we can do is take each slice of the network and add new links. So there are now connections from one node in one layer to the same node in another layer. If I bring those links to be very, very, very small in weight, I'm essentially just clustering separately each one of the layers, right? They're independent. And if I dial up those links all the way and make them maximally important, then I just get these like long strings where I connect one node to itself all the way throughout the multi-layer network. But somewhere in between, interesting things start to happen, right? I can get clusters that now bridge multiple layers together. And some of the most interesting work on this was originally done looking at voting data in the US House of Representatives and Senate, where you can see not only voting blocks in particular years, but voting blocks sort of extending throughout time. And as you vary this inter-layer strength, you can sort of vary the resolution or vary like the focus on your glasses with which you're seeing these voting blocks over time. Super cool analysis. There are generative approaches as well. One idea is to write down a stochastic block model for each layer. But we can jointly model layers whenever their structures are sufficiently similar. So it could be that I have one block model for this layer, and then another block model that produces 3 layers, and another block model that produces, let's say, the remaining 5 layers. So in this way, I'm sort of compressing what would be a large number of layers into a smaller model. Another generative approach is to have a stochastic block model for each layer. And we're going to model all the layers simultaneously with the same community structure, the same division into groups, but allow the block matrix to differ. So allow the relationships between the groups to vary. So let's say like when we're in this room, we're all in one group. But when we're outside of this hotel, we're now in a different sort of block structure where, let's say, even though we were connected within the room, outside of the room, we're disconnected. One is in general preferred if the nodes appear and disappear over time, and you're interested in clustering things in time. Whereas I think these generative models, 2 and 3, are preferred if you want to solve what I'll call the layer interdependence problem. So if I want to learn about which layers are related to which other layers, we can solve that problem. So are 2 layers structurally similar? Are they complementary? Are they neither? What we can do is learn a stochastic block model from m layers, and then try and predict the lengths of the m+1 layer. This tells me whether or not those m layers taken together provide a good model, a good description of the m+1 layer. If I do a totally bad job at predicting those other lengths, then they're not a good model. And if I do a very good job, then they are a decent model. So one example here is from a social support network across 2 villages in South India. And there are 12 different layers, and they're asking questions like, who'd you go to if you need somebody to look after your kids? Who'd you go to if you needed a little money for the bus? Or what if you needed a more substantial loan? Who might you ask? As it turns out in this case, as we add more layers, our prediction quality goes up. So the yellow triangles here, you can see doing okay about prediction. As I now add 2 layers, L equals 2, I get these green or gray boxes. My prediction quality goes up. 3 layers, prediction quality goes up again. And if I use all 12 layers and I just hold out a fraction of the edges, and I try and predict that fraction, I do the best. So we're not showing the details here, but you can actually diagnose which layers in particular are going to help you predict which other layers, suggesting that those layers are closer together in sort of the fundamental mechanisms that are creating the patterns that we see. So more layers in this case means better performance. So the layer structure was generated by sort of the same social mechanisms. Probably we shouldn't be surprised if you trust somebody to do work in your house, you probably trust that person with your children more likely than some other random attribute. So we tried it with malaria as well. And something interesting happens, which is that we do pretty well if we try and predict with a single layer. And now it's the opposite case. As we add more layers, the prediction quality gets worse and worse and worse. Which is to say that as we add more information from the different pieces, and then we try and predict, the reason that we're doing worse is because the sources of information are conflicting with each other. They're telling us different things. And in retrospect, this is something that we should expect, right? The layer structures have been generated by different biological mechanisms because they're trying to evolve independently. So in this case, the multi-layer approach tells us something that in some ways we already knew, which is that the parasite is really optimizing to be able to move each of these layers independently. And just because I have information about 4 of the layers doesn't mean that I have any better information about the 5th. In fact, if I try and generalize the lessons from the first 4 layers to predict the 5th, I do worse and worse. So some key points to wrap up here. We talked about generative models. These are powerful statistical tools. They can perform rigorous inference of network structure. We talked mostly about community structure, but we can also learn about hierarchies through these latent space models. We talked about single layer, multi-layer, bipartite, and so on. And the key points from this were points about model data mismatch. When we have a model for the data, and it's the appropriate model for the data, then we do well. And if it's the wrong model, then we don't do well. So generative models strengthen weakness together. Talked a bit about multi-layer networks, and these are very fresh results. And if you start looking at some of the papers on these. You're going to see everything published in the last 5 years. So this is really the edge of what we know. And I expect in the next 5 years we'll know a lot more. These are great when there are multiple link types. When we talk about versatility, which are the key interlayer connectors? Robustness, how can we make networks resilient? Or, the flip side, if we want to dismantle a network, which are the nodes we should attack? We talked a little bit about interdependence, how the layers actually relate to each other, and how we can use one layer to predict another. And then finally, tying it all together was malaria. And I think there are just beautiful network problems here presented to us by these miserable parasites. But the networks have been very helpful. They've shown us that there's this hybrid evolutionary strategy. We want to maximize diversity while at the same time preserving function. And finally, the networks showed us that there's no layer interdependence, that these genes really are evolving separately. This was something that trees couldn't tell us, but luckily these network approaches can. So I'm happy to answer any questions. The PDF slides that you'll see online will have references in the first 2 slides, so any of the links or papers you want to follow up on are there. But of course, you can always email me as well. So, thank you.